Today we're going to focus on biochemistry. Biochemistry, in this chapter, we're going to address what is water, the components of water, and the different attractions. And then we're going to move into organic macromolecules such as carbohydrates, proteins, fats, and nucleic acids. Now let's break apart the word biochemistry. Bio, meaning life, chemistry is referring to how the materials, the chemicals interact. So we're going to put them together and we're going to address how this is going to be more on a atomic level, more of a microscopic anatomy. So we're going to be focusing on how these chemicals interacting together will help us sustain our life. So this is going to provide energy as our body uses these molecules. Let's do a little review first. We have inorganic versus organic compounds. Remember, inorganic, we did already address what minerals were. Inorganic compounds don't have carbon, specifically carbon, hydrogen, and oxygen together. Inorganic would be something that is not living or not part of something that was living or created from something that was living. This is going to be like water. There's no carbon with hydrogen, with oxygen there. It's just hydrogen and oxygen. Different salts like NaCl, so sodium chloride, potassium chloride. Many of these salts don't have carbon in them. Many acids and bases. Then if we contrast that with an organic compound, we can see that it has carbon. It has carbon, hydrogen, and oxygen together. And we're not talking about CO2 because, yes, we have C and O, but that's not considered organic. We need the carbon, hydrogen, and oxygen together, and that's going to be what we're going to really address today with the carbohydrates, lipids, proteins, and DNA and RNA. As we address water first, water is the most abundant chemical in our body. We said our body is made up of over 60% of water. Now the molecule of water or compound of water is very small, and because of its design and the size that it is, it's able to move very fast. It's able to go through very small spaces as we maybe talk about osmosis and diffusion. Hydrogen has a positive charge in the molecule while oxygen has a negative charge. So they're kind of like opposite poles, kind of like North Pole, South Pole, or the positive and negative of a magnet. That allows one part of water to be attracted to the other part of another water molecule. So hydrogen would be, which is positive, would be attracted to the other water molecule, the oxygen part. Now this attraction keeps them together. So you probably remember in chemistry class that water in and of itself, the H2 bonded to the O, that H and O that's bonded together is a polar covalent bond. So it is going to be more of a stronger kind of shearing bond. But when we're talking about a water to a water, the hydrogen and the oxygen being bonded together, that's going to be a hydrogen bond. So that's going to be more of a weaker bond. So it is easier because of the hydrogen bond for it to break apart. So it is easier for the water to break apart other charged molecules and dissolve them. And water is called the universal solvent. And a solvent means that it does the dissolving. So it's able to break these things down, able to dissolve many solid particles. Now you do need to know about, because of the polarity of water, water is able to stick to itself. So the attraction between the hydrogen and the oxygen, that hydrogen bond, that is going to be called cohesion. So water attracted to water. But water can also stick to other materials, so something different. So it's adding something new, adhesion. So it's sticking onto your skin or sticking onto the side of a bucket. So it slowly goes down. It's sticking. That attraction is called adhesion if it's something new, something different, something other than water. But when you have water to water, that's cohesion. That's what the cohesion is, what gives it its dome and its water droplet shape. And specifically on the outside of that droplet, that's going to be called surface tension. So the stronger attraction on the outside of the droplet is called surface tension. Water is also considered a buffer. Water is usually, if you are drinking just regular water, maybe like a tap water or bottled water, that's going to have a stable pH of 7, so a general pH of 7. If you do drink more of a carbonated water, it's going to be more acidic. So water can kind of go either way. Normal water, because it's in the center there for the pH, it can be kind of acidic or basic, but it's generally more neutral. Water also has, for heat capacity, water can absorb and release heat energy slowly. So that's why maybe something solid, the sand, would be hotter than the ocean, the water. That's why your swimming pool may take longer to heat up than a metal object or a solid object. And water can hold a great deal of heat energy. So even though it may take longer to heat up, it's going to hold that heat energy longer. It helps the marine animals, the fish, and the other organisms living in the ocean maintain their body temperature at a safe range. Okay, if you ever heard of thermal pollution, that's why if you put an animal in from something warm to something cold too fast, that fish or that animal can easily not adjust and easily die. Now let's move to macromolecules. Macromolecules, we talked about, are very large molecules. So if we have a molecule or a compound and they keep on assembling, get bigger and bigger and bigger, you have a large chain, then that's going to be called your macromolecule. Macro meaning large or big. 4 types of macromolecules you're going to need to know: carbohydrates, lipids, proteins, and nucleic acids. You can pause it, read through these. I'm going to talk about these in more detail in just a little bit. Let's get into more detail about carbohydrates. We said those are generally considered sugars. Carbs are sugars. There's lots of different sugars. There's fruit sugar, bread sugar, and we'll talk about all those. Milk sugar, and we'll talk about all those in just a little bit. But we say carbohydrates are organic. And so what we're going to be talking about from now on in this lecture is organic materials. And generally the ratio for carbohydrates is going to be about 6 carbons. And you can go through and look at these pictures and kind of count them out. Generally you're going to have 6 carbons. You're generally going to have double the amount of hydrogens. So about 12 hydrogens. And then usually you have about 6 oxygens. Now some of these you're going to have less oxygens than others. So you can go through and kind of count the O's on some of these. And if you look at glucose, you're going to have 6 oxygens. But if you go down to ribose, it's going to have 5 oxygens. But deoxyribose, if you count that only has 4 oxygens, because deoxy meaning less oxygen than ribose. So they do differ a little bit, and for each one of these different sugars, they generally have the same amount of elements, but they're going to be reshuffled or changed. You can see some of them are a hexagon shape. Some of these are a pentagon shape. And so when we have a hexagon shape, we call that a hexose. When we have the when we have the last 3 letters OSE, OSE stands for a sugar. So this is a hexose, so a hexagon sugar. When we have pentose, that's a pentagon sugar. So basically these are all monosaccharides. These monosaccharides provide a source of energy to our cells. So basically your digestive system breaks the sugar down, the carbs down, the bread, the milk, whatever it is, has our blood carry it to our cells, and our cells in the cytoplasm and the mitochondria break down the sugar to make energy. And this is going to be in a process called glycolysis and the Krebs cycle. And we're going to learn about that later. So what we're going to have here is glucose, which is a basic simple sugar, hexose shape. Then we have fructose, which is a fruit sugar. Generally, you're getting it from fruits and vegetables. That's a pentose sugar. Then we have galactose. That's not a very common one, but galactose will bond to form a milk sugar, and we'll talk about that in just a little bit when we learn about disaccharides. Then we have a deoxyribose, which is the DNA sugar, and then we have a ribose, which is the RNA sugar. Now these are all basic compounds, but then when we bond them together, so when we bond glucose and fructose together, we release oxygen. This is called a dehydration synthesis because we're getting rid of oxygen to bond them together. Synthesize to bond together. We form a sucrose. Now, sucrose is generally a plant sugar. This is found in many nuts and fruits and veggies. Primarily, when you hear of white sugar, that's going to be your sucrose as well. Then let's look at the bottom. We bond glucose and glucose together. We get a maltose. Maltose is basically a grain or bread sugar. This is found in cereals and starchy foods like sweet potatoes and fruits. And then we have your galactose, which we were just talking about, and glucose. Galactose and glucose make milk sugar. So that's going to be your lactose. If you ever heard someone is lactose intolerant, means they're having trouble breaking apart this lactose bond. So this is found in dairy products. And then now we're going to focus on polysaccharides. Poly means many. Remember, mono meant one, di means two, di or bi means two, and poly means many. So this is a polymer, a large linking together of different molecules, different compounds of the same type. And so basically we have many simple sugars here that are bonded together. So it's not just one, it's not just two, it could be hundreds. And what we have here is some examples. Starch. Starch is a main example of a polysaccharide. Okay, then we have cellulose. Now cellulose is a structure it's a plant sugar that is in the cell wall of plants. It gives the plant its durable strength and structure. So basically that's why we can't get a lot of nutrients from plants if we're eating it raw. Cows and other animals will chew the cud. So they're going to chew the plant, swallow it, and then throw it up again, chew it again until they are able to break down the plant. And that breaks apart these strong, large bonds. Then you have glycogen. Now, glycogen is specifically in animals, and for animals, fungi and bacteria. But since we're talking about humans, we're going to focus more on glycogen than the other two. And this is going to be more of a sugar, a carbohydrate that is stored in the skeletal muscles and the liver. If you ever heard of blood glycogen, especially people who have diabetes, where their body may not be producing enough insulin or their insulin's not working properly. So if they have too much blood sugar or blood sugar levels. So this is going to tie in with diabetes. Now iodine, which we talked about in the previous chapter, iodine. So iodine is usually a brownish orange color. So when you drop the liquid iodine on bread or potatoes, it will turn from a brownish yellow orange to a dark blue, a blue, purple, blackish color. So it will signify, yes, there is starch present. Now let's move to lipids. Lipids, just like your carbohydrates, have carbon, hydrogen, and oxygen. It is organic. Lipids, however, have two kind of structures here. They have a kind of head structure with a kind of a tail structure. And so what we have here is for neutral fats, we have neutral fats which are also called triglycerides. We have a glycerol head, and then off of that we have three fatty acid tails. And when they do bond, we have a synthesis reaction. Three waters are given off to make that neutral fat, that triglyceride. So think about neutral fat, triglyceride, three. So three fatty acid tails. Now, lipids, specifically neutral fats, triglycerides, are going to be found in your blood. They can be stored in your fat cells. They can be converted into calories. They could be stored around your organs, your lower skin between your muscle and upper skin layers. Hormones are made up of lipids as well, and hormones can release triglycerides for energy. The second type of lipid is a phospholipid. So it's going to have three parts now: a phosphate head, a glycerol, and two fatty acid tails. Now, fatty acids hate water. They are hydrophobic, so they're going to move away from water. Now, the phosphate head is going to be hydrophilic. It loves water. So hydro meaning water, philic meaning love, phobic meaning hate or afraid of. So what's going to happen is they are going to line up, especially in this construction or the formation of the cell membrane. The cell membrane makes a phospholipid bilayer. Let me show you a picture. So here's the cell membrane, and basically you have the phosphate heads and then you have the fatty acid tails. Those two fatty acid tails are hydrophobic, remember? So they're going to try and point away. The hydrophilic phosphate heads are going to be on the outside touching the water. That's why if you ever have grease, sometimes you have these grease clumps, because the phosphate heads are going to be on the outside, the hydrophobic tails are going to be on the inside, making a separate clump. That's why they separate from others. Then we have steroids. Basically, steroids are found in cholesterol, different body salts, vitamin D, and sex hormones, as well as adrenal hormones. And steroids primarily have a flat structure with four interlocking hydrocarbon rings. So you can kind of see those four rings there. Three of them are a hexagonal shape. Now, as we're talking about cholesterol, which cholesterol is a type of steroid, there is three types of fats that you should be aware of. There's saturated fats, unsaturated fats, and trans fats. Now, the best one, or the one that you're encouraged to eat the most or have, is an unsaturated fat. Now, unsaturated fats are basically monounsaturated fats, monounsaturated fats, and polyunsaturated fats. Remember, mono just meaning one, and poly meaning many, so it's larger. Unsaturated fats are primarily going to have one or more double bonds to those carbons. And so basically you have fewer hydrogen atoms on their carbon chains. The key here is that these double bonds are weaker than the single bonds. These unsaturated fatty acids, these unsaturated fats, are going to be more of a liquid at room temperature. So they have a lower melting point. And these are primarily found in a lot of plants, plant oils, fish foods, and fish oils. So fish meat. These are going to be your unsaturated fats. So a lot of nuts, a lot of plants, a lot of fish. A saturated fatty acid is going to have a single bond. And this is primarily going to be in meats and dairy. And here's the problem here is that a saturated fatty acid, a saturated fat, is going to be more of a solid at room temperature. So the fear is because they have more of a low density lipoprotein, this is going to yield a higher cholesterol level. And what can happen is because it's more, or it can be solid at room temperature in your body, it can start solidifying and blocking the arteries. So the saturated fats can cause cholesterol to build up, especially a lot of fried meats, a lot of dairy. It's going to build up in your arteries, and it can increase the risk of heart disease and stroke. Now, not until more recent, we're going to be addressing trans fatty acids or trans fats. Now, trans fats are artificial. These are manufactured. So these are going to be fats that no longer the FDA deems safe. These fats, just like saturated fatty acids, these fats can be solid at room temperature or semi-solid. And basically, these are in a lot like your Twinkies, your cookies, your foods that need to be preserved so they last longer on the shelf of the supermarket. Now let's move to proteins. Proteins are made up of amino acids. Now, proteins can be 2 amino acids. It can be a combination of 20 different types of amino acids bonded together. Now, as they're bonded together, they're going to be bonded together by a peptide bond. And that's something you will need to know. What bonds and holds the 2 amino acids together to form a protein? It's a peptide bond. Now, if we look at the structure of each amino acid, and as those amino acids do bond together, we learn that this is called synthesis, coming together, and we're going to give off water, okay, through that process, dehydration synthesis. If we break apart the protein into 2 amino acids, then it would be called hydrolysis. That's the other direction. But let's take a look at the amino acids. So we have the amine group, and that focuses on nitrogen. Nitrogen, 2 hydrogens. We have a carbon with a hydrogen, and then that R represents a carbon chain, many carbons. And then we have the carboxyl group, the C double bond O, OH. Now, you probably learned last year about enzymes and protein denaturation. Enzymes are proteins that are comprised of amino acids. And basically, enzymes are going to interact with maybe other proteins or other materials such as carbs or fats. Now, the enzyme has an active site. This is where the substrate, what you want to break apart or what you want to put together, attaches to the enzyme at the active site. And basically, the substrate, which we said could be another protein or fat or sugar. And in this case, in the picture, we're synthesizing it. We're building it together. So we're going to lose water, and we're going to bond it together, and we're going to bond it together if they are proteins, amino acids, by a peptide bond. Then that protein separates from the active site of the enzyme, and that protein, that substrate, leaves. Okay, so in that picture, that is going to be dehydration synthesis. So synthesis is when you have 2 separate ones come together at the enzyme and it yields as one. Now, digestion, we talked about that before, hydrolysis. This is when we have 2 things being made. So we start with 1. So the substrate is maybe a macromolecule, maybe a disaccharide, maybe a polysaccharide. The enzyme bonds with that disaccharide, that substrate at the active site, and then with water breaks it apart. Now we're going to focus on the nucleic acids. And basically the structural unit of the nucleic acid is the nucleotide, which is composed of nitrogen, a nitrogen base, like adenine, thymine, guanine, cytosine, a pentose sugar, so os meaning sugar, and this is a pentagon, five-sided. The carbons make up the little angles or the little corners, and then it has a phosphate. So deoxyribose, this deoxyribonucleic acid is going to be called DNA. And this is a double-stranded helical, so it's spiral, and it's kind of like one side is going up, the other side is going down. It's kind of like opposites. It's inverted. It's kind of like an inverted type of ladder. This makes our genes. This is stored in the nucleus. It provides instructions for making proteins. And then RNA, or ribonucleic acid, has one extra oxygen in its sugar. That's why DNA is called deoxyribo, because it has one less oxygen, deoxy. So going back to the ribonucleic acid, it's usually single strand. Sometimes it can be kind of balled up, but basically it doesn't have thymine, it has uracil. So let's look at some pictures, some visuals for these. So this is your basic kind of structure of DNA. You have a phosphate, sugar, and then nucleic base. That's one side of the ladder. The other side of the ladder is going to be the other nucleic base that bonds with that one. A always bonds with T, adenine always bonds with thymine, and then guanine always bonds with cytosine. And then we have another sugar and another phosphate. But you can see that sugar and phosphate are kind of upside down, so they're kind of inverted on the other sides. Now RNA, we said, is going to be, RNA will not have T, but RNA is going to have U instead. U instead. So it's going to have adenine, guanine, cytosine, and uracil. So they're going to bond the same way. So G always bonds with C, but in this case, A, we don't have a T, A is going to bond with U. And there are three varieties, three types of RNA. There's the messenger RNA, which is copied from DNA in a process called transcription. That mRNA is going to leave the nucleus. It's going to leave the nucleus after transcription. It's going to find the ribosomal RNA. Do you remember learning in 7th grade ribosomes? That's what a ribosome is. It's just a ball of RNA. So we call it a ribosomal RNA. It's kind of like a train station. The mRNA is kind of like the train. It comes into the train station, into the ribosomal RNA, and it waits for the people. And the people are the tRNA, the transfer RNA. Now, as the transfer RNA, the red, yellow, pink, those three little prongs, that could be an A, a U, or G. It could be a U, a G, or a C. When they come in, they're going to bond with the corresponding nucleic bases in the train station, in the ribosome. So as they bond, as the tRNA bonds with the mRNA at the rRNA, it's going to make a protein. It's going to make an amino acid. So you learned probably in 7th grade that ribosomes make proteins. This is going to be how. And we're going to talk about transcription and translation. That's what this is. When the mRNA bonds to the tRNA at the ribosome, that's called translation because we're making something new. We're kind of translating it from the mRNA to proteins. So that's going to be translation. So we'll learn about transcription and translation in a future chapter. Here we can see and take a look at the DNA and contrast that with the RNA. And here you can see DNA to DNA, we're making more DNA parts, that's replication. But then when we have the mRNA kind of rewrite the DNA, that's transcription. That happens in the nucleus. The mRNA then leaves the nucleus, goes to throughout the cytoplasm, finds the ribosome, bonds with the ribosome, which then comes into the ribosome, which then bonds with the tRNA, mRNA with tRNA at the rRNA. That's called translation because it makes proteins or amino acids. Now, this is the last thing we're going to talk about is ATP, adenosine triphosphate. How many phosphates? Tri, so that's 3 phosphates. ATP is just an energy molecule. We're going to be learning about this when we talk about glycolysis and the Krebs cycle. So this is going to be happening at the mitochondria. ATP can be produced in the cytoplasm as well as the mitochondria, but this is going to be primarily your energy source. Please remember, we have triphosphate, 3. Now, when we start producing energy for our body, we're going to start breaking apart the bonds of phosphates. And then these can be reassembled, storing more energy for another use.